Through constant innovation, Baxter supports your ICU's needs with a broad range of extracorporeal organ support therapies. From CRRT to sepsis management and beyond, we are your dedicated partner, helping you save and sustain lives.
Hello everyone, I'm happy to welcome you to this session of the second World Sepsis Congress. We have a very exciting session on update on adjunctive sepsis therapies, and I'm happy to welcome all the audience from more than 150 countries. Our sponsor for this session is Baxter, that we wish to thank for his participation. Our first speaker will be Jean-Louis Vincent. He is a professor of intensive care medicine at the University of Brussels. He is the president of the World Federation of Societies in Intensive and Critical Care Medicine, and has been the president of other societies such as the European Society of Intensive Care Medicine, the European Shock Society, the International Sepsis Forum. He is the author of more than 900 articles and 400 book chapters in review. And of course, he is very well known in the field. And especially also everyone is knowing is an international symposium on intensive care and emergency medicine that is organizing for more than 38 years by now in Brussels. So Jean-Louis, happy to welcome you and listen to your talk on recombinant human thrombomodulin.
Thank you very much, Jean-Marc. It's indeed exciting to address this topic. As you can see from this first slide, the management of sepsis has three components: the infection control, the hemodynamic stabilization, and what we will speak about now is the use of thrombomodulin as part of the modulation of the host response. We know that the endothelium is a very important target in sepsis and other acute diseases, and there is an interaction between the coagulation system and the inflammatory response. This has been shown in a number of studies, and since our time is limited, I will move on and indicate that activated protein C was or is a very important player in this field, and we could show already 17 years ago that activated protein C can reduce mortality rates in septic shock. And very interestingly, we could have a good explanation as we understood more and more how activated protein C works. Unfortunately, another quickly performed study was totally negative, and the so-called Xigris was taken out of the market. Nevertheless, there is a sound rationale for its use, and we could show with these instruments allowing to evaluate the microcirculation that activated protein C could improve the microvascular perfusion. So this was a very important step. We thought that we could go and actually target the endothelium through the coagulopathy, which is present. And in this sub-study, it was shown that patients with severe disseminated intravascular coagulation benefited more from activated protein C than the others. If I'm mentioning this, it is because thrombomodulin obviously acts in concert with protein C, and they do act on the endothelial protein C receptor. Thrombomodulin also has other activities including on the complement pathway, and it also interacts with high mobility group B1, HMG-B1. So it's not just an anticoagulant as heparin could be. There are very good experimental studies showing that thrombomodulin can protect animals from organ dysfunction. And this is true also in sepsis. This is true also in ischemia reperfusion of the kidneys, for instance. Now, if we quickly progress to the clinical data, there are good data indicating that the plasma soluble thrombomodulin levels are associated with worse mortality rate in sepsis and ARDS. Now, for a while, a Japanese company has prepared a recombinant human soluble thrombomodulin called Recombinant or ART-123. And they did studies in Japan on patients with disseminated intravascular coagulation, showing that it may actually reduce mortality rates in this patient population. Now, the data may not be entirely convincing to all of us, but they led to the commercial availability of recombinant thrombomodulin in Japan. So we already have some interesting observation from the use of thrombomodulin in septic patients in Japan. And using propensity scores, a number of multi-center studies have shown that thrombomodulin administration may shorten the hospital stay, lower hospital all-cause mortality, and provide a longer survival time. Interestingly, the bleeding complications do not seem to be a big problem. Now, in all these studies, and this could be anticipated, the sicker the patient, the greater the benefit. And if we go back for a moment to the PROES study with activated protein C, it was the same observation. Patients with a high Apache 2 score were more likely to benefit. So again, this is a recent study showing that anticoagulant therapy with thrombomodulin can result in lower mortality rates, especially in the sickest patients. So we recently wrote this editorial saying that there may be a place for this type of compounds in patients with severe coagulopathy, especially when it's associated with sepsis, as shown here in this meta-analysis. The higher the risk of death, the greater the benefit with recombinant thrombomodulin. So with this in mind, we did a study in patients with sepsis and coagulopathy. Let me first quickly underline that in terms of pharmacokinetics, hopefully, we do not need to adjust the doses very much in patients on renal replacement therapy, and apparently not in patients with moderate liver dysfunction as well. So that's something which is worth mentioning. Now, we need to better characterize the patient populations, and as I wrote here in the recent paper, we should not do prospective randomized control trial on two heterogeneous patient populations. That's how I do not think that sepsis therapies should be looked for. We should look for specific subsets of patients who may benefit from this particular intervention. That's what we mentioned in the JAMA, for instance. We need to look for specific patient populations, and that's what we can call personalized medicine, maybe not precision medicine, but personalized medicine, which is much better than the poorly characterized patient populations that we have studied for so many years with negative trials in sepsis. So we have a number of ways to characterize the septic patient, and since with thrombomodulin we target the coagulation system, I think it's appealing to go for a disseminated intravascular coagulation due to sepsis. And in the first study, it was a phase 2B study, we reported 5 years ago that there may be a survival advantage in patients with severe coagulopathy and respiratory or cardiac dysfunction. That's how we went on and did another large study, which is a phase 3 study now, including 800 patients, and the study was completed recently. So we enrolled patients with infection, organ dysfunction, cardiovascular or pulmonary, and a coagulopathy characterized by a prolonged INR and thrombocytopenia. Now, we can only show you the first data because they were part of a press release. Further data will be presented at the European Society meeting in Paris next month. But as you can see on this slide, there was some reduction in mortality rate. Being 29.4% in the placebo group, it went down to 26.8% in the ART-123 group. You may say that the difference is only 2.6%, which is actually not meeting the criteria we choose for positivity. But I think personally that we are in a gray zone here. We cannot say that the study is negative. We cannot say that the study is positive. We are doing some further analysis on subgroups of patients based on the severity of coagulopathy, based on the type of organ dysfunction, based on the timing of intervention, based on the severity of the disease. Clearly, we will need additional data. But I think the data are quite positive in a way. They are even exciting because we cannot expect too much from these new therapies when you think at what can be really influenced by a new therapy. So here it is a 10% reduction in mortality rates. If we look at 29%, going down to 26.8%. So this is not totally positive, but it's not bad. And so the nice thing, I believe, to finish up, is that this may be valuable not only in sepsis, not only when there is an infection, but in patients showing signs of coagulopathy and organ failure associated with DIC. So it's possible that in the future, a signal could come from the lab telling us that the patient has these alterations very easily recognized in any hospitalized patients. So to conclude, there is an urgent need for new sepsis drugs. The importance of coagulopathy is well established. Endothelium is a valuable target. There is a wide availability of simple blood tests to identify the suitable patients. And here we have a study now showing some reduction in mortality with this intervention. So there is more to come. Stay tuned, and we will have more data presented in Paris next month. Thank you.
Yeah, regarding the better way to treat the patient with a personalized medicine and finding subset of patients. So the ART-123 study, was it including that type of characterization of the patients, or it were just introduced as septic patients?
No. As I said, it was infection plus coagulopathy, as reflected by a prolonged INR and a low platelet count. So we needed to have these criteria present to enroll patients in the trial. So that's how I refer to personalized medicine. Indeed, we didn't want to just enroll patients with sepsis. I think that's over. We should no longer do that. And I know I'm preaching the convinced because you, Jean-Marc, have done these studies on this.
Absolutely. And also, you mentioned the sicker is the patient, the better is the effect. So do you think as well that clinical score should be taken into consideration for the inclusion of the patients?
That's a good question. We could either take a score or we could have organ dysfunction criteria. As you know, in some studies, they took either an Apache score or a SOFA score. Probably the SOFA score is even better because the Apache score takes into account elements like age, presence of cancer, which indeed influence outcome, but may not necessarily influence the response to a new intervention. So we already wrote several years ago an editorial in Critical Care Medicine saying that we should not use an Apache 2 score to refine the patient enrollment. We should rather select them on the basis of organ dysfunction, and that's what the clinician would like to do at the bedside. We don't want to calculate a score, but simply look at the presence of organ dysfunction.
Okay, Daniel, Jean-Louis, I think our time is over, so I thank you for your presentation, and I think we need to move on for the next speaker.
Excellent, thank you.
Thank you, Jean-Louis. The next speaker is Peter Piekers from the Department of Intensive Care Medicine in Radboud University in Nijmegen in the Netherlands. He's Professor of Experimental Intensive Care Medicine, has co-authored more than 300 articles and 25 chapters, and to have visited the place, I know that he has a very efficient specific design unit to study the endotoxin in human volunteers, and this is providing a bunch of great information in human volunteers. So, Peter, are you with us?
Yes,
if this is okay, excellent. You will talk about the alkaline phosphatase.
Yes, thank you so much, Jean-Marc, and welcome to all the listeners that are online. I will talk about alkaline phosphatase as part of the STOP AKI study, the sepsis trial of alkaline phosphatase in acute kidney injury patients. So actually, we know that from sepsis research that, for example, gram-negative bacteria contain endotoxin, and this is actually recognized by immune cells, and so the whole pro-inflammatory response is started, and pro-inflammatory mediators are released. And actually, as a collateral damage, this might induce organ dysfunction and even death in septic shock patients. Now, the interesting thing is that endotoxin contains two phosphate groups, and if you would remove these phosphate groups from the endotoxin, it's no longer active anymore. And that's why alkaline phosphatase becomes interesting, because we know alkaline phosphatase as a measure for liver dysfunction, but we should realize that it does so by detoxifying toxic compounds. So alkaline phosphatase is also able to remove phosphate from, for example, endotoxin, and thereby the endotoxin is no longer pro-inflammatory, but actually an antagonist. And so if this pro-inflammation is inhibited, maybe the whole inflammatory cascade and the deleterious effects in organ function and survival are also influenced. Actually, there are two small phase 2 studies that were conducted with bovine alkaline phosphatase, following a lot of animals research that showed beneficial effects. And in these trials, it was actually shown that indeed patients with sepsis that were treated with alkaline phosphatase showed a more pronounced improvement in their endogenous creatinine clearance, as well that their need for renal replacement therapy tended to be lower, and also the duration on renal replacement therapy was lower. However, these were very small trials, only 36 patients per trial, and this was done with a bovine alkaline phosphatase. But based on these results, the company actually decided to develop a human recombinant alkaline phosphatase. And what they did is combine the placental form of alkaline phosphatase with a very long half-life in the crown domain, and the intestinal alkaline phosphatase that is very biologically active in the catalytic domain. And so the recombinant was a combination of both, and so that we would have a very active alkaline phosphatase, but also a very long half-life. Well, the safety was shown in phase 1 studies, and then the trial in septic patients with AKI was designed. So this trial consisted of 2 parts. First, in part 1, there was a dose-finding part with 3 doses of alkaline phosphatase against placebo, and then in part 2, in a blinded fashion, a DSMB decided what was the optimal dose, and part 2 was conducted with that optimal dose. What we looked at was, again, creatinine clearance from day 1 till 7, but also renal replacement therapy needed by the patients up to day 28, and also day 60 and day 90 follow-up, especially relating to mortality and major adverse kidney events. What we did in this study is the usual inclusion and exclusion criteria. Patients, of course, had to provide informed consent or their next of kin. These were adult patients. They were allowed to have sepsis for 4 days, no longer than that, because we want to have a clear relationship between the onset of the sepsis and the onset of the AKI. And also the AKI should be there not longer than 24 hours to improve the possibility to have a beneficial effect of a therapy. The exclusion criteria were similar as seen in previous trials, so we don't want any pregnant HIV-positive drug-abusing leukemia patients in this trial, and all the other exclusion criteria are very common as well. So the trial was built as 2 parts, and as you can see in this slide, in the first part, the 3 doses and the placebo contained approximately 30 patients per group, and then following on, the optimal dose was chosen by the DSMB. It turned out to be the highest dose. Nobody knew that while the trial was conducted, and this dose was then continued, and all the patients that were included during this phase contributed to the primary endpoint of efficacy as well. Well, of course, in this trial, it was a randomized and blinded trial, so it turned out that the groups were very good comparable, as you can see in this table 1. And you have to believe me that they were very similar, actually, except for a few important things. It turned out that the renal function in those patients that received placebo was a little bit better compared to the alkaline phosphatase treated group. These are small differences, but we think later on that this might be of importance, and that's why I am highlighting it here. So the interim analysis with all the 3 doses groups and the placebo group showed that in the placebo group, the average endogenous creatinine clearance over the first week was 46 mils per minute. And actually in the 0.4 it was a similar number. In the 2 high dose groups of alkaline phosphatase, it was 63 in the 0.8 milligram group and 60 in the 1.6 milligram group. So based on also on this efficacy and also on adverse events, the DSMB decided that the trial should be continued with the highest dose. And important to emphasize that this was still blinded to all study personnel. Then for the treatment emergent serious adverse events and all adverse events, the safety was very important of this first inpatient trial. Again, a lot of numbers in this table. Please, you don't have to look at them in detail. What I can show you is that the number of adverse effects was similar in all groups. There was no signal of any dose dependency in any specific side effect, and also no formation of antibodies of any relevance was found against this recombinant alkaline phosphatase. So you have to trust me on that. Then next, the efficacy. What we did find in the first week was that the increase in endogenous creatinine clearance was not significantly different between the active group and the placebo group. And also, the key secondary endpoint, renal replacement therapy incidence, was not significantly different between groups. So the primary endpoint was clearance over 7 days. This was exactly the same, and I will illustrate here in more detail. So no therapeutic benefit of this compound. However, we did look for longer-term effects as well, and we measured creatinine clearance up to day 28. And if that data is also included, the whole curves become significantly different, as you can see here. And this is based on the difference in creatinine clearance on day 21 and day 28. So this is a benefit on longer-term renal function by recombinant alkaline phosphatase. Then the survival curves. This is the Kaplan-Meier curve of those patients that were treated with placebo, and on day 28, 26.7% of these patients died. In those patients that were treated with alkaline phosphatase, mortality was significantly lower, and on day 28 it was 14.4%. This was a significant effect, and also up to day 90, this significant signal was still there. So have a look at the major adverse kidney events on the longer term. No difference between groups on day 28. On day 60, there was a significant benefit in the active group compared to placebo. And also on day 90, the MAKE composite was significantly improved in patients that received recombinant alkaline phosphatase. And this difference on day 60 as well as on day 90 was mainly driven by the difference in survival. So because this strong and unexpected effect on survival, we did additional exploratory analysis. And what we did was we did a multivariate analysis in which treatment was included, but also, for example, other organ dysfunction, so SOFA scores per point. And as you can see, the hazard ratio is still significant if we have these other factors included in the model as well. 50% reduction of the chance to die with alkaline phosphatase, while SOFA scores, if they increase, naturally there's an increase in mortality as well. And we also did it in another way, a forward stepwise multivariate analysis in which we had significant predictors of bad outcome, like a package score of the clearance at baseline, and still the treatment arm, so treatment with recombinant alkaline phosphatase, was beneficial to these patients, and it remained significant. And as you can also see in the lower part of the slide, is that we found that there was a very important relationship between the baseline creatinine clearance with a median value of 40 as 0, and that those patients with a lower baseline creatinine clearance had a higher chance to die. And so the small difference between groups might be of importance there. We looked at the pre-specified subgroups, and we found no effect of baseline inflammatory markers or baseline tubular injury marker levels, and no effect of time between the diagnosis of AKI and the start of the treatment. We did find an effect of renal function at baseline and the treatment effect. So as you can see, if you can see here, those patients with a baseline creatinine clearance of more than 60 had a much better prognosis than those with a baseline clearance of less than 60. And if you look at the therapeutic efficacy of alkaline phosphatase, there was no therapeutic efficacy in those patients with a good renal function, with a clearance above 60, while in those patients with a baseline creatinine clearance below 60, the beneficial effect was most pronounced. But all these are post hoc exploratory analysis just to try to understand how this survival benefit would work. So in conclusion, recombinant alkaline phosphatase therapy was considered safe and very well tolerated. There were no specific side effects and no higher incidence of side effects. The treatment did not affect short-term renal improvement or renal replacement therapy need in 28 days. However, the longer-term renal function from day 21 and day 28 was significantly improved by recombinant alkaline phosphatase, and also long-term major adverse kidney events, and survival was significantly better in those patients that were treated with alkaline phosphatase. And with that, I thank you for your attention, and I'm happy to answer all your questions.
Sure. I mean, thank you, Peter. Because of short time, I will just take a question from the audience. And one of our guests was asking what were the other drugs which were given to the placebo and to the treated patients.
Yeah, so these were patients in the clinic with septic shock, mainly in the ICU. So this was an on-top treatment. So patients were treated just as standard care is done right now. All the treatments that were given to all our patients, and the alkaline phosphatase was given on top of that standard of care.
Okay, just another question from myself. Are those patients have been selected for being positive for endotoxemia?
No, they were not. Actually, one thing is, and I gave it as an example, that the endotoxin has phosphate groups that can be removed and it's no longer toxic, but also other compounds and circulating toxins have phosphate groups. And we have now shown in animal studies that, for example, ATP is also toxic and pro-inflammatory, and the alkaline phosphatase is able to remove phosphate from ATP, and ultimately adenosine is formed, which is anti-inflammatory and tissue protective. So we now think the mechanism of action is not only dependent on the endotoxin pathway, but also on less specific and also on other toxins that are there and that also contain phosphate groups.
Okay, excellent, Peter. I thank you very much for your presentation and your interesting data. I think we need to move on, and I'm happy to welcome Pierre-François Laterre. He's the chief of the intensive care service at St. Luke University Hospital. Is very much involved in shock ARDS. Is a member of many national and international societies. Is the author of more than 180 papers, and he will talk us about adrenomodulin. So Pierre-François, we are ready to listen to you.
Okay, you hear me, Jean-Marc?
Yes, I do, like everyone around the planet.
Okay. Thanks for the introduction. So my short presentation here, my goal is mostly to show what the development is ongoing regarding using adrenomodulin or antagonizing adrenomodulin in the context of septic shock. And so adrenomodulin is something that plays an important role in the maintenance of the vascular permeability or protecting the endothelium. So what is known in sepsis in general is that there's significant damage of the endothelium and an increased permeability, at least in the patient showing the biggest severity. And one of the peptides playing a role in the vascular tone and permeability is adrenomodulin. And it's a 52 amino acid peptide that is considered as vasoprotective. So on the one hand, it plays a role in maintaining the vascular integrity, but at the same time, when it diffuses through the vessels and goes to the muscle and the smooth muscles, it induces a vasodilation. So it's a kind of paradox because this hormones expressed and produced mostly by endothelial cells and smooth muscle cells as a kind of double action. But at least what has been observed and known already now is that there's a nice correlation in between the circulating level of adrenomodulin and the importance of hypertension, and there's a good correlation between the need of vasopressor and the level of adrenomodulin. Also, elevated adrenomodulin levels have been associated with an increased mortality. And we've looked at this as others, and we found that the cutoff value of 70 is clearly associated with a dramatic increase in mortality going above 35% at day 28. And so what is believed and known, at least based on experimental study, is that adrenomodulin, when it's staying into the endovascular space, plays a role in protecting the vascular integrity and reducing the permeability. On the other hand, when it diffuses through the vessels and go into the interstitium, it induces vasodilation. And so therefore it explains the importance of correlation between adrenomodulin level and the severity of shock, but at the same time, adrenomodulin is important to protect the vessel. So this kind of balance is a challenge, of course, for a drug, because you need to have adrenomodulin staying in the vessel and acting to protect the vessel, and at the same time, we want to prevent from hypertension. And this is how the antibody, so adrenizumab, is being developed. This antibody fixed to the N-terminal of adrenomodulin, but does not block the action of adrenomodulin into the vessels, and so still is expected to protect the endothelial barrier and reduce the permeability. And so when you look at the complex of antibody together with ADM, adrenomodulin remains active and is able to fix to its receptors on the endothelial part of the vessels. So this large antibody at the same time remains into the circulation and does not diffuse into the interstitium. It has a long half-life, since the half-life is 15 days, and the development and the study done experimentally have demonstrated that when you inject the antibody staying in the vessels, it's able to fix ADM and capture part of the ADM located in the interstitium, but remains bioactive within the vessels. And you see that on the right part of the figure here, that there is a nice correlation in the activity of ADM in the circulation according to the dose of antibody given. So it shows that you are removing the ADM from the external part of the vessels into the intravascular compartment, and there the ADM is still active on its receptor. This has been done also in healthy volunteer, and this work comes from Peter Spiegel group, where according to the dose of antibody, you see that the bioactivity of ADM in the circulation increase. And once again, this is not due to a change in the synthesis of ADM, but clearly to the fact that you're removing the ADM from the extravascular compartment to the circulating level. Now, in terms of efficacy of the antibody, it has been studied in animals, and here in this model of CK ligation in mouse, the administration of the antibody against adrenomodulin has improved the kidney function, the requirement in fluids, the requirement in noradrenaline, but has also modulated apparently the inflammatory response, and there is a reduction, at least in the circulating TNF and IL-6 in these animals. I was dealing with the experimental data that have been published a couple of years ago, and some of them are very recent, showing that the use of the antibody against adrenomodulin in a second model of 2 hits in pigs, at least, hemorrhagic shock followed by sepsis, was associated, interestingly, with a reduction in the need of fluid to achieve the goal of hemodynamic control, and much more animals had a rapid improvement in their shock as opposed to the animals treated with vehicles. Now, of course, after the experimental data, what has been, of course, studied is the tolerance of the antibody in healthy volunteers, and this is a work from the group of Peter Pickers, where in a phase 1A, healthy volunteer received the antibody, and this were well tolerated, whatever dose used. And in the second part of the study, so the phase 1B, the healthy volunteers were receiving a continuous infusion of endotoxin for 3 hours, and after 1 hour, they were given the antibody against adrenomodulin. Of course, endotoxin in healthy volunteers is not supposed to induce shock, but still they feel sick. And interestingly, what has been seen is that the healthy volunteer receiving the antibody felt more rapidly better than the one receiving the vehicles in terms of the sickness score, as this can be observed in this kind of model. Now, after this, it was ready to start a phase 2 trial, and this has been initiated in March this year. And the goal of this study is to enroll patients with septic shock having an associated elevation of ADM. So there is a bedside assessment of adrenomodulin in these patients with shock, and if they fulfill the criteria of shock together with elevated adrenomodulin, they are eligible for the study. And we plan to enroll 300 patients in a 1-1-2 randomization design. One arm with a low-dose adrenomodulin antibody, a second arm with a 4 milligrams per kilo antibody, and a third arm with placebo. And currently we have enrolled already 50 patients, and we hope to complete the study by the end of next year with the 300 patients. And the main objective is not only safety, well, secondary endpoints include the more rapid improvement in shock, the duration of shock, and the change in the SOFA score over the first 2 and 7 days with a penalty associated with mortality. So just to conclude at this point, so the adenomatuzumab is a non-neutralizing antibody against adrenomodulin. It stays in the vascular compartment and maintains the activity of adrenomodulin, but prevents from accumulating adrenomodulin into the interstitium, acting as a vasodilator. So it maintains vascular integrity based on experimental data. It is safe in healthy volunteers, even challenged with LPS. And we hope to support by the phase 2 the demonstration of a benefit in terms of shock and confirm the reduction in permeability and the reduction in fluid in these patients. Thank you.
Thank you very much, Pierre-François. We have to move on because of the time. I'm very sorry.
Okay.
But thank you for this very exciting new piece of information. Our next speaker is Richard Hotchkiss. He's from the Washington University School of Medicine, St. Louis, Missouri. Of course, he's a very well-known investigator in the field. He was the one who demonstrated the problem of apoptosis of human cells in patients, and clearly his laboratory has been on the forefront to propose new immunoadjuvant therapy for this disorder. So, Richard, happy to welcome you.
Thank you, John Mark. I'm very glad to be here. So I'm going to try to update us on some new approaches to sepsis, including the results of 2 new recent clinical trials. There's my acknowledgement slides. So as many people know, sepsis has been considered the graveyard of pharmaceutical approaches. That is, there have been over 50 trials of different anti-inflammatory and anti-cytokine agents to treat sepsis, and they failed uniformly. This led to a reevaluation of the concept that deaths in sepsis were due to uncontrolled systemic inflammation. I like this slide. It kind of says what I feel sometimes. Just because the path is well trodden does not mean that you're heading in the right direction. So what I'm going to try to convince you in the very brief time here is that in sepsis there's a new paradigm that both pro and anti-inflammatory responses occur early in sepsis, and that as sepsis continues, there is relative immune suppression. That is, the patient's immune system is not functioning normally, it's impaired. That patients who have this immune suppression may die due to either their inability to control that primary infection, or frequently what happens is they develop new secondary hospital-acquired infections. Immunity on these patients remains impaired for months, and sepsis is the most common cause for readmissions to the hospital. All right, here. So a new way to treat sepsis would be to target the host immune system. That is, when you look at who are the patients that are dying of sepsis, in general, it's patients who are elderly. These patients have what's called immunosenescence, just meaning their immune system is not functioning well as you age. Other patients that tend to die from sepsis are patients with a lot of comorbidities, patients who have underlying liver disease or kidney disease, and patients who are alcoholics. These are the patients that tend to die. And the reason that they die is because this population of patients have are well known to have weakened immune systems. So it's logical to target those patients to boost immunity to improve outcomes. Boosting immunity could also decrease their incidence of hospital-acquired infections. And the other important thing, if you are able to increase the host immune system, is you will protect them against a broad range of pathogens. There are these new emerging superbugs that are very frightening, and these usually occur in patients with weakened immune systems. So if you could give agents to boost the immune system, you are likely to improve outcomes. I would also point out that boosting the immune system, as people know in the oncology field, in cancer, that this has been the breakthrough, a transformational. There have been over 100,000 patients, many more. It's changing the dynamics in patients with cancer. And it is the most exciting area, as indicated by the ability of a significant percentage of patients of cancer to now survive, not just go into remission, but to survive. So this has been a topic, as indicated here, in the New England Journal. If you do look at the literature and follow medicine, the New England Journal has highlighted the fact that by boosting the host immune system, particularly focusing on improving the T cell, the lymphocytes, that is the current new paradigm. Now, why am I talking about cancer when I should be speaking about sepsis? The reason is because cancer and sepsis share many common immune suppressive mechanisms. We have discussed this as a well-recognized fact that when investigators have examined what mechanisms, that is, what are the common causes of immune suppression in patients with sepsis and cancer, they see that they share many same mechanisms. For example, the checkpoint inhibitors, blocking PD-1, it works in both infectious disease and in cancer. There's increased T regulatory cells in both. There's a production of the anti-inflammatory cytokine IL-10 in both. The reason for this is because of the receptors, the TLR receptors that mediate the response of the immune cells are promiscuous, meaning that they are activated by a variety of different types of mediators and antigens. And you have these mediators released both in cancer and in sepsis. So this, as I mentioned, the checkpoint inhibitors are leading the way in this. President Jimmy Carter, who had metastatic melanoma to the brain, was treated with these drugs, and is now out 2 to 3 years. It's a fairly that's unheard of. It was uniformly fatal a few years ago. So what did the checkpoint inhibitors do? The checkpoint inhibitors release the brakes on the T cells. The programmed cell death ligand 1 interacts with programmed cell death 1 on the T cell, and that is a mechanism that the body has to down-regulate the T cell. When you have an infection, your T cells are upregulated. And so there has to be a way for the body to turn off the T cell, and that is what PD-1 does. So these checkpoint inhibitors, the anti-PD-1 or anti-PD-L1, which are now clinically approved for 8 to 10 different types of cancers, take the brakes off the T cell and let it function again. That's the best way to look at it. This is demonstrated most dramatically in a case I was involved with, published in the Lancet Infectious Disease, a 30-year-old female who was a victim of that Brussels terrorist attack, who suffered a number of bone injuries as well as intra-abdominal injuries. She was incredibly ill. She arrested in the emergency room. She had infections with multi-drug resistant pathogens, as well as a thing that was really causing the most serious problem was mucormycosis, an invasive fungal infection. After a number of weeks, the invasive fungal infection, the mucor, was involving the spleen and the stomach, and she went to the operating room and those organs were removed. However, the mucormycosis was also involving the great vessels in her abdomen. This was not able to be resected. You can't do that. So in desperation, the physicians treating her measured her immune function and showed that her immune function was suppressed. And based upon the fact that her immune cells had increased expression of this programmed cell death 1, they initiated treatment with these checkpoint inhibitors. Nivolumab, which is anti-PD-1, and interferon gamma were started on a compassionate basis. And the patient slowly resolved this infection, which invasive intra-abdominal infection, really, if you're not able to resect it, is pretty much uniformly fatal. So this was felt to be, she survived and was discharged from the hospital. A major evidence showing that these immune checkpoint inhibitors could be used in infectious disease. Based upon that, as well as a number of animal studies, a checkpoint inhibitor trial was initiated, supported by Bristol Myers Squibb. It was a multi-center trial, and the conclusions to get to several trials. As a quick summary, the checkpoint inhibitor was well tolerated in the patients with sepsis. There was no evidence of the checkpoint inhibitor anti-PD-L1 causing a worsening cytokines. The cytokines were not exacerbated. No worsening fever or shock. So this was the primary we were looking for was safety, and it was shown to be safe, and there was evidence on a laboratory basis that it improved immune function. Another trial that was just recently begun was done with interleukin-7. IL-7 has been called the maestro of the immune system because it works on CD4 helper cells, which are the major T helper cells that orchestrate the global immune response. As you know, in HIV, it's the loss of the CD4 helper cells, and they are critical in coordinating the immune response. What previous studies from our group, as well as the numbers, have shown that when you obtain different lymphoid organs, including spleen or other organs, from patients who died of sepsis, and you compare them to patients who died of non-septic causes, and then we did staining for these critical immune effector cells, the CD4 T cells and the CD8 T cells. And what you can see is that there is a profound loss of the CD4 and CD8 T cells in the spleens and other organs from patients who died of sepsis. In fact, they look very similar to patients with HIV in that regard. And this has been well documented both in adult and pediatric and neonatal sepsis. So theoretically then, interleukin-7 could be helpful because it is a cytokine growth factor and increases the number of lymphocytes in response to its therapy. As I've said, IL-7 has been used. It's been a favorite of many investigators at NIAID and in the NCI to treat immune deficient patients. This was a study conducted by Levy, who showed that there was a dramatic increase in CD4 and CD8 T cells, a 3- to 4-fold increase beginning within the first week after starting IL-7 therapy. we, in conjunction with Bruno Francois, Thomas Ramelli, as well as Ed Sherwood at Vanderbilt, began a trial of IL-7 in patients who had lymphopenia, had low lymphocyte counts in patients with sepsis, and those results were recently published in JCI Insight. It was one of the few trials to target to enhance. The goal was to enhance immunity, and is the first therapeutic trial to work on improving the adaptive immune function. There were 27 patients in a double-blind, randomized, placebo-controlled trial, and we were looking for safety and ability to increase CD4 and CD8 T cell counts. There were 2 different dosing regimens of IL-7, as well as the placebo group. And what did we find out? The absolute lymphocyte counts to be entered into our study, the septic patients had to have a very low absolute lymphocyte count. And the placebo-treated patients, if they survived, had a slow increase in their absolute lymphocyte counts. The interleukin-7 was dramatically effective in increasing the absolute lymphocyte counts, which are measured on your complete blood count in the laboratory, by 3 to 4-fold. Importantly, it was a persistent increase. In other words, the lymphocytes stayed elevated for several weeks after stopping therapy. And so theoretically, the IL-7 could improve immune system, a sustained improvement to prevent these secondary infections. Shown here is the fact that the IL-7 increased both CD4 and CD8 T cell counts, so it had broad effects on immunity. The drug was well tolerated. The only adverse effect was a grade 2 to 3 rash. It also had effects we determined to increase the function of the T cells, help them to fight the infection. In conclusion, sepsis evolves into this immunosuppressive state. The patients that die from sepsis in general are patients who are elderly, have comorbidities. These are the ones that have the much higher incidence of morbidity and mortality. The immunologic defects in sepsis are very similar to those occurring in cancer, suggesting a common mechanism, and that immunotherapy that's working now in cancer may also do so in sepsis. We do believe that will be the future of infectious disease. It will be to investigate the defects in the patients that have sepsis, and you'll have targeted drugs to improve the different functions of that. Interventions which with the potential to improve immune function are being studied now with a number of trials. I would like to thank my sepsis team, the IL-7 team, Bruno Francois, Thomas Dax, Robin Genet in Lyon, Thomas Ramillon, Guillaume Monterey, Vanderbilt, Ed Sherwood. With our anti-PD-L1 trials, Elizabeth Colson and Dennis Grasella from BMS, Derek Angus, Sachin Yendi, Link Moldauer, Craig Cooper-Smith, and Elliot Krauser. Thank you very much, and I'll be happy to answer any questions.
Thank you so much, Richard. Because we are already behind schedule,
Sure.
I'll still transmit two questions we get from the audience. And one of our guests asked, how early do you recommend treatment with immunotherapy? And he also asked, do you think it's beneficial to a subgroup of patients? And this may be linked to another question I got, which is monitoring immunity needed to allow using immunotherapy?
Yes, those are great questions. Those are critical questions, and they're spot on. We did not begin immunotherapy until after 36 to 48 hours of sepsis, so that we believe, and there's data, that the patients move from the hyperinflammatory phase to a more predominant net immunosuppressive phase over the first few days. The data on cytokines tend to show that. So there are patients, however, that, like if you have a lot of comorbidities or elderly, these people have depressed immunity. They're walking around at baseline with depressed immunity, and they could probably treat it very early with the onset of sepsis. The secret is to develop tests to immune phenotype the patients so you know what condition their immune, either in the suppressed or in the hyperinflammatory phase. So, so that sort of gets to both questions. The secret is a good test to tell the status of the immune system that will allow you to safely. We did use a low absolute lymphocyte count that had to be low and persistently low, and we believe that is one marker for immune suppression.
And I have a very brief question regarding the anti-PD-L1 treatment. Is it affecting apoptosis?
We did not look for it in our septic patients, but there are a number of trials in animal studies, and yes, it is pretty good at being anti-apoptotic and blocking the sepsis-induced cell loss.
Okay, thank you very much, Richard. It was a great talk. Thank you, as usual. And because of lack of time, we'll need to move to the next speaker.
Okay, thank you. I appreciate it. Bye-bye.
Bye-bye. So the next speaker is Ashish Khanna. Is an intensivist working at the Center for Critical Care and at the Cleveland Clinic Laterre College of Medicine in Cleveland. Is the Vice Chief for Research for the Center and the Quality Director for the Cleveland Clinic. And so we are happy to welcome him and to listen about his presentation on Angiotensin II.
Thank you. Thank you, Dr. Kavalian. It's a pleasure and an honor to be a part of this World Sepsis Congress. I'd also like to thank Dr. Simon Finfer, Flavia Machado, and Conrad Reinhardt for this opportunity. And good morning, good afternoon, and good evening to whatever part of the world you all are tuned into as you listen to this next talk. So I'll be talking about Angiotensin II, and this talk comes at an interesting time because we're almost a year or more out from the publication of the results of the Angiotensin II in high output shock phase 3 trial. And we're almost 6 months or more from when the drug was approved by the US FDA for use in septic or other distributive shock. So we are at a stage where there is clinical use of the drug available and also some interesting subgroup data that I would like to share with the audience today and really shed some light on how Angiotensin II is beneficial as a adjunctive therapy in sepsis and septic shock. So the problem that we're going to talk about today is refractory vasodilatory shock, and septic shock can most certainly evolve into a refractory vasodilatory shock, which very simplistically speaking is just hypotension that persists despite the use of high doses of vasopressors. Now, the definition of high doses of vasopressors has not been set to a certain standard. Various investigators have described high doses of vasopressors as anywhere between 0.2 micrograms per kilo per minute to all the way up to 0.8 or even 1 microgram per kilo per minute of norepinephrine or norepinephrine equivalents. But what is very clear in literature is that exposure to high-dose vasopressors is bad, in that 30-day all-cause mortality ranges anywhere between 50 to 80% in patients who are exposed to high-dose vasopressors and who are faced with a refractory vasodilatory shock syndrome. The vasopressor toolbox that every intensivist has as of now is largely composed of a lot of catecholamines, norepinephrine, epinephrine, norepinephrine being our go-to drug to treat hemodynamic instability in vasodilatory shock or septic shock, followed by vasopressin, and now, with the inclusion of the renin-angiotensin-aldosterone system and angiotensin II, this vasopressor toolbox seems to be more complete, and there is certainly another go-to agent in the armamentarium, which has a separate mechanism of action from catecholamines and vasopressin. The important thing to remember about the usage of vasopressors in septic shock or vasodilatory shock is not to rely on monotherapy. Monotherapy has been shown to be toxic at high doses, and especially monotherapy with catecholamines. Angiotensin II itself is certainly not a new agent. It has reemerged after being quiet for almost 6 decades or more. In the year 1957, a group of scientists in the United States, and interestingly at Cleveland Clinic, and also a group of scientists in Latin America came together and initially reported the successful synthesis of angiotensin II. At that time, this agent was actually called hypertensin. And the initial case reports and case series of the use of angiotensin II were very optimistic, very favorable, but somehow in the next 5 or 6 years after it was first synthesized, it never really got a marketing or pharmaceutical industry support for production, and it kind of vanished, only to be resurrected, say about 6 decades later, when in 2014, Lakshmi Chawla's group at George Washington University first published the ATHOS trial, or the angiotensin II in high output shock trial. And this was a pilot study. It was a 20-patient study that really showed a signal to benefit, where human synthetic stable angiotensin II increased blood pressure and had a catecholamine sparing effect in patients with high output shock. This led us to conduct the phase 3 placebo-controlled randomized double-blind multicenter study of a compound LJPC501, or human synthetic stable angiotensin II, in patients with catecholamine-resistant hypotension, and that is what was the ATHOS-3 trial. The ATHOS-3 trial was done under a special protocol assessment agreement with the US FDA, which really meant that the United States Food and Drug Administration looked at the protocol and was in complete concurrency with the primary and secondary outcomes and the trial design. What did ATHOS-3 showed? So ATHOS-3 essentially showed that angiotensin II led to a hemodynamic outcome, which was the primary endpoint, of a MAP of 75 or 10 more than baseline at 3 hours from the initiation of drug infusion in about 70% of patients treated with angiotensin II, compared to 23.4% patients treated with placebo, a highly significant outcome in favor of angiotensin II. The blood pressure response to angiotensin II was very robust, and a significantly higher blood pressure was achieved in patients receiving angiotensin II in the first 3 hours, and a consistent and stable difference was seen in the angiotensin II and placebo arms all the way through to hour 48 of the required drug infusion. Further, an important secondary efficacy endpoint that was a consequence of the primary endpoint was that a cardiovascular SOFA score benefit was seen in the angiotensin II arm compared to the placebo arm. And a clinically very relevant additional endpoint was that patients exposed to angiotensin II had a significant decrease in background vasopressor usage, or a catecholamine unloading effect that was significantly seen more in the angiotensin II arm compared to the placebo arm. The angiotensin II molecule led to a significant catecholamine unloading, and this unloading was highly significant in the first 3 hours of the study drug infusion, and a separation in the requirement of background vasopressors was seen all the way out to hour 48. A question that was asked was survival after initiation of therapy. So mortality as an outcome was an exploratory outcome for the ATHOS-3 trial. It was a small trial, 344 patients, and certainly not a trial that was designed to test for survival or for a survival benefit. We did see an initial trend to a improved survival with angiotensin II, but certainly this did not translate into statistical significance. Trial results were published in August 2017 in the New England Journal of Medicine, and we have been almost a year out since then, and the world is obviously asking a question of what to do with angiotensin II and where does it fit in terms of adjunctive therapy in sepsis and septic shock. So let me go over some subgroup data that's going to shed some light on how angiotensin II is valuable in sepsis, septic shock, and refractory vasodilatory shock. The first interesting data set is a survival benefit looking at angiotensin I and angiotensin II ratios. So the enzyme cascade is renin, angiotensinogen, angiotensin I, angiotensin II, and then angiotensin II having its downstream effects secondary to aldosterone and direct effects. We looked at a population median in the ATHOS-3 population. Population median of an angiotensin I to angiotensin II ratio was 1.63. We divided the population to a less or higher ratio. And what we saw was that in patients with a high angiotensin I to angiotensin II ratio, which means lower endogenous angiotensin II, in those patients, exposure to exogenous or synthetic angiotensin II led to a survival benefit. Now, this is a very interesting outcome and very relevant because a lot of our current population coming into emergency rooms, operating rooms, and ICUs walks around with 15, 20 years of ACE inhibitor therapy behind them. And maybe this is a interesting patient population and a relevant niche for angiotensin II when these patients have refractory or other vasodilatory shock. The other interesting outcome that has been published so far is outcomes in patients with acute kidney injury, knowing that septic AKI is a cause for morbidity and mortality in these patients. We looked at patients in the ATHOS-3 cohort who developed acute kidney injury after initiation of angiotensin II or placebo, and we saw that patients who were exposed to angiotensin II had a survival probability which was almost which was to the tune of 53.2% compared to 29.6% in placebo. Again, a significant survival benefit in patients with AKI exposed to angiotensin II. In these patients, we also saw that their hemodynamic profile was better and they were liberated faster from renal replacement therapy if they were on angiotensin II. The other interesting question that has been asked is, how do I know if my patient is going to respond to angiotensin II? Now, in the ATHOS-3 trial, the drug was started at 20 nanograms per kilo per minute, and the investigator could up-titrate the drug up to 200 nanograms per kilo per minute. In some patients, a hyperresponse was seen, in which case the blood pressure was extremely high at standard doses of angiotensin II, and the investigator had the option to down titrate the study drug to less than 5 nanograms per kilo per minute. We looked at these hyperresponders to see if they had a survival benefit. And we saw that comparing patients who were on less than 5 nanograms per kilo per minute of angiotensin II with higher doses at 30 minutes after drug initiation, patients who were on the lowest dose of angiotensin II had a survival benefit that was significantly more than patients on higher doses. And this was even after controlling for a big set of disease covariates of all sorts. So an interesting outcome that almost tells the clinician that if your patient has a really robust blood pressure response on initial exposure to angiotensin II, and that response comes quickly and at the lowest possible dose, then this is a patient that probably has a physiological need for angiotensin II compared to the other patients that are, unresponsive or slow to show a response. So where does angiotensin II fit in our treatment paradigm? Well, shock or vasodilatory shock or septic shock, there is no going away from identification and treatment of cause, source control, antibiotics, surgery, whatever it takes for appropriate source control, necessary fluid resuscitation, and always norepinephrine to go as your first line treatment of the vasopressor side of things. But once there is inadequate hemodynamic control with norepinephrine, the message to the audience is early initiation of either vasopressin or angiotensin II, rather than waiting for a higher dose of norepinephrine monotherapy, exhausting catecholamines, and then moving on to your second or third agent. The message is synergism of vasopressors, because truly all three mechanisms need to be acting in synergy for appropriate blood pressure management. And in addition, the other adjuncts, as in steroids, as in ascorbic acid, thiamine, even the likes of methylene blue, need to be implemented earlier rather than later, because the earlier you intervene, the better outcomes you would see in hemodynamics and overall outcomes of patients with sepsis, septic shock. Thank you, and I'm open for questions.
Thank you, Ashish. I think because, again, lack of time, I will just transmit one question we get from the audience. And the question is, how do you feel about replacing norepinephrine as first-line vasopressor in favor of angiotensin II or vasopressin, given the biological deficiency of vasopressin in sepsis patients?
Well, even though angiotensin II is highly potent and the initial data is very favorable, I would still not recommend angiotensin II as your primary vasopressor. I think norepinephrine makes physiological and pharmacological sense. It's easy to titrate, and it has been a drug that's been in use for almost 60, 70, 80 years, and the data is way more than the available data on angiotensin II. So even though angiotensin II is a very potent vasopressor, I would restrict it for vasodilatory shock that reaches a point of hemodynamic instability and is difficult to manage with norepinephrine versus using it as a first-line agent.
Well, thank you so much, Ashish. I think we need to conclude.
Thank you.
It was a great session. Also, I would like to thank the chairs of this Congress, Flavia Machado, Simon Finfer, and Conrad Heyer, for putting all together this great meeting. I would like to invite the audience to visit the website, to sign the World Sepsis Declaration, and to follow all the activities on Facebook, Twitter, and Instagram. And also would like to thank Baxter for supporting this session. With that, I think I can close the session. Thank you very much.